we present UKBOB 1 billion MRI labeled masks for generalizable 3D medical image segmentation. Joint work between me, Abdullah, Emmanuel, and Amir from VGG Group at University of Oxford. SAM, or Foundation Model for Promotable Image Segmentation, paved the way for generalized image segmentation models, but it is labor intensive, requiring extensive prompting, and does not generalize for 3D image segmentation. MedSAM fine-tuned SAM model on a large collection of datasets on medical images, showing great generalization on medical images, but it still struggles with 3D context for 3D segmentation, and it still requires heavy prompting to work on 2D image segmentation. Recent developments in SWIN Uniter, a transformer model specialized for efficient 3D medical image segmentation, showed great promise for efficient 3D segmentation that generalize very well, and this architecture is considered one of the strongest architecture for 3D medical image segmentation. This also coincided with the developments over the years in the UK Biobank, which becomes the largest MRI dataset in the world right now, with over 50,000 MRI samples that have extra metadata information associated with it. In this work, we present UKBOB, UK Biobank Organs and Bones, which is a segmentation dataset that's the largest in the world, based on the UK Biobank MRI dataset. UKBOB comprises 51,761 3D MRI scans from the UK Biobank and over 1 billion 2D segmentation masks covering over 72 organs. The size of the circles represent the 2D image resolution, and as you can see from the blue circle, our UK Biobank Organs and Bones, or UKBOB in short, is the largest in terms of diverse medical image dataset available. It has 72 organs, as mentioned, labels, and covers 17 million 2D images. Here is an overview comparison between different 3D medical image datasets, and as you can see, the UK Biobank is orders of magnitude larger in terms of number of samples, but also in terms of diversity. The dataset collection procedure starts from the UK Biobank, and then we annotate the UK Biobank using automatic annotation using the Total Vibe Segmenter model, which was released earlier this year. This model was trained on 51 hand annotated labels of body organs from NACO and 15 subjects from the UK Biobank based on the NNUnet. We use this model to automatically collect the labels for the entire UK Biobank samples with 72 organs. Here we show examples of the kidney or organ, for example, shown on the label on the right. However, we cannot verify the quality of these automatic labeled or pseudo labels collected for the UK Biobank, so we propose several ways, mechanisms to verify that. One of them is that there was a previous study that collected manual annotation of spine for spine curvature analysis, and we found the spine accuracy to be 81% in the UK Biobank organs and labels compared to the ground truth manual annotated labels, which indicates that the labels are pretty accurate, as can be seen here from the visualization. We also collect our own manual labels of subset of UKBOB, which is a 300 samples of UKBOB manually annotated, and the precision of the original or the big UKBOB is 87.3 in die score
on the manual collected subset.
We filter out this data and labels using normalized statistics by normalizing by the body volume since some bodies are different, and we remove extreme epsilon samples that are beyond predetermined threshold.
In addition to the normalized volume, we also use the sphericity and eccentricity feature of each organ class to statistically flag out inaccurate labels. The combination of normalized volume, sphericity, and eccentricity statistical filter gives us the SOLF filter or specialized organ label filter, which achieves the best precision in cleaning the collected labels.
To validate this, we use a set of experiments and data sets that are used as validation, like the BTCV abdominal CT scan dataset that has abdominal organs that intersect with the UK Biobank organs and labels. We use also BRATS for 3D medical image segmentation for tumor segmentation, and AMOS for abdominal MRI organs as well. Here we show that the zero shot performance, that means training a model on the UKBOB labels and then testing directly on these AMOS and BTCV dataset, we can see that the effective filtration on the downstream zero shot generalization uh, that used to verify the effect of the filtration. Here we saw the distributions of the labels before and after filtration. We also resort to entropy test time adaptation since these pseudo labels created have some noise associated with them. So the idea of this entropy test time adaptation is to refine the batch norm layer of the network we use, which is the SWIN uniter, um, at test time according to the entropy map. So these entropy map can be created from the prediction, so that you don't need ground truth. Using the network prediction and the softmax probabilities of the network for each label, you can create these entropy maps, and these entropy maps after that can be used to refine the batch norm um, layer of the network, and hence you can adapt the network for each test sample. This adds an extra boost to the performance of any network compared to another test time adaptation baseline across different datasets. Here we show the results of the different networks trained in the UK Biobank and tested on the validation set of the UKBOB dataset. You can see that the SWIN uniter achieved the best results, and this model that's trained on SWIN uniter on UKBOB, we call it SWINBOB, and this is our foundation model proposed in this work. Here we test the SWINBOB model on BTCV dataset, showing great generalization on CT scans, even though it is trained on UKBOB and then fine-tuned on BTCV. The fine-tuned model on BTCV achieves state-of-the-art results on the standard benchmark for abdominal CT scan organ dataset. And when we test our model and fine-tune it on BRATS 3D tumor segmentation on the brain, we also, our SWINBOB model achieves state-of-the-art results with significant improvement compared to previous models. One of the most interesting charts about this work is how can we make sure that the large data set we collected has value, which is known as the pre-training scaling law. So as we increase the number or the size of the pre-training data set, which is the UKBOB, we measure the downstream fine-tuning performance of the SWIN-BOB network on BTCV and BRATS. And you can see that there is a linear increase in the performance compared on the downstream tasks as we increase the percentage of the pre-training dataset of the UKBOB dataset from 20% to 100%. This confirmed the utility of the large scale pseudo labels that we collected on the UKBOB dataset. Here we show a Tisney visualization of the features of the Swin Bob model, and you can see that different classes have unique distinct groups which indicate the utility of these features from the Swin Bob and the UK Bob dataset.